Good morning, everyone. We would like to thank Dr. Jane Bai for graciously accepting the invitation of PAASE to speak before this group and other interested members of the faculty, students in the Philippines. Dr. Jane Bai is with the U.S. Food and Drug Administration as a regulatory review scientist. She's working on quantitative systems pharmacology for regulatory drug development and for assessing biomedical products. So she has been with FDA since 2005. She has co-founded and co-chaired FDA System Pharmacology Scientific Interest Group since 2015. She also served at the NIH Links Consortium Steering Committee and is currently a member of NIH NIBIB. Dr. Bai received her master's degree in applied mathematics and her PhD in pharmaceutics from the University of Michigan, Ann Arbor, Michigan. She was also a faculty member of pharmaceutics at the University of Minnesota. For several years, she was an entrepreneur exploring biotech ventures, and she received state and NIH funding. She licensed in silico software. It's a very recent development in silico. So she has licensed this software to pharmaceutical companies, and the software is Aura Spotter, and it predicts oral drug absorption in humans. In addition to publishing about 70 peer-reviewed papers and book chapters, she co-edited a book entitled Systems Medicine for the Methods in Molecular Biology series, which is for publication in May by Springer. So let us welcome Dr. Jane Bai.
I'm sorry, I forgot to unmute myself. Thank you so much for nice introductions. And now I would like to share my slide. Please kind of let me know whether you can see my slide. Can you see my slide, please?
Yes.
I would like to first thank you. Thanks PAASE for the invitation, Dr. Lau, Dr. Domingo, and Mario for their invitations. And I would like to thank Dr. Ron Quiro for a very nice presentation, and also my colleague who has been very helpful, and also my previous ORISE now is an associate professor at University of North Dakota, and also USDA for the data on magnesium absorption. So I would like to first introduce the concept of system biology. System biology is an approach in biomedical research to understanding the larger pictures, be it at the level of organism, tissue, or cells, by putting the pieces together. It is in stark contrast to decades of reductionist biology, which involves making the pieces apart, taking the pieces apart. And this is a definition I took it from NIH. Now, starting from for a computational model, we would add cell biology, and then we will move on to adding proteomics informations, genomic informations. Certainly that had to with biological context, and it makes sense to be biological meaningful. And certainly we don't want to forget about immunology, because when we have a disease manifestation, adult disease, immune system is involved. And put them all together, forming a computational modeling, network analysis. Now, if we put a drug in there and to include mechanism of actions, the pharmacological pathway from drug target to pharmacodynamic response that can be measured using analytical methods through quantifying biomarkers or imaging responses put together, then we have a system pharmacology model or system pharmacology network. In terms of computational approaches, we could use different mathematical computation to link drug target to the PD effect and to predict efficacy or even predict off-target effect in terms of safety. And today I will cover both. And so this is a graph showing a drug. Once the drug is taken either orally or by non-parental administration, the drug getting into the body through absorption, such as oral absorption or transdermal absorptions or other routes or subcutaneous, drug will be absorbed and then distributed throughout the body, and then will be metabolized by the livers or some of the enzyme in other tissue, but mainly by the livers, and then eliminate through secretion through the kidney. Now, in terms of pharmacokinetics, we understand that the drug molecule distributes throughout the organs and distribute to the heart, kidney, liver, brain through the circulations, and that's where the drug can reach the target site. Now, for example, in the liver, that not only when it reaches the liver, this organ, but then it distributes into the cells, and there are different type of cells in the liver. This graph is to show you that when we consider system multiscale modeling, we will consider all of scale down to the cells, such as couple cells in the livers or lymphocyte, or other cells. Then we will further consider organelle in individual cell types, and to be able to link molecular pathway and the drug, the dose together to predict response. So starting from a drug molecule, a dose, and then down to the cell organelle levels and molecular level, put everything together to understand drug effect. So this is another scheme showing that how we can utilizing the information we have accumulated throughout the past 40, 50 years from the drug that have been approved or drug that did not reach the market that was discontinued. We can, based on their interaction with the protein in the cells, we can set up and based on our knowledge of biology, we can put in the pharmacological pathway together, so we will be able to utilize the data available from post-marketing or from the drug that have been discontinued, but link them in a way to their target protein-protein interaction, pharmacological pathway, and then link it to, in terms of the pathway, the effect in individual organs. And then we will be able to link to such as adverse reaction, the phenotype. And so this is a system level, the system integrations, so from drug protein interaction to protein pathway interaction through to protein organ interaction and organ phenotype interaction. And certainly we can be as detailed as possible. We have done mathematical computation using linear integer programming, or we can set out ordinary differential equations. This graph is to illustrate that we can utilize big data. NIH have so-called I2B2. They support several consortium to utilize gene expression data and proteomics data, or even a cell level readout in terms of different measurement of the cell response. So we can have this utilizing the information from that have been accumulated. We can, based on the chemical universe of the drugs, and link it to genomic information and knowledge. We will have the targeted disease population that can utilize those informations. And then at the cell level, we can use so-called transcriptomic data, a 3D micro systems such as organ on the chip, or the gene expression data from animal. We know Japan used to have, and I'm not sure that is still active, that they have a data base based on the for liver toxicity that they use animal, I believe it's rat or mouse. And then we can also utilize human tissues. At the system biologists in Seattle, System Biology Institute is well known. And one of the expert there, Dr. Abede, is a lead food that advertising 4P, that is how to utilize individual person's information throughout a lifetime and to monitor and to help advise individual patients, individual subject in terms of the health and how to guide that person to work, to live longer and have a healthy life by monitoring that person information, including genomic omics data. And so that is a very different scale, but this is to say that we can integrate information out there, data out there that have been accumulated through our research worldwide, including United States and Philippines and other countries. We can integrate the data together and try to predict for drug effect or toxicity and to do a drug repurposing. Most recently, organ rare diseases, FDA have an initiative, ACR, how to accelerate cure for rare ARC for cure, to accelerate cure, finding a cure for rare diseases. So that is our recent initiative. So it's to utilize publicly accessible data and knowledge available for us to do research and to make use of the knowledge and data available. Here, I just want to list the databases that I have been using, and I have to apologize for a lot of text here, but if you're interested, just check it out, and I'm sure there are a lot more databases and knowledge bases out there now, especially in terms of modeling, multiscale modeling. NCI recently actually have a FOI, that is a funding announcement. They solicit professors or researchers to curate the database and the model that have been published to use them for research to find a better cure for treating cancers. So this is the continued list of the information and knowledge base that are available for us to to put data available to do the mining and computations. Now, I would like to use a drug-induced retroanalysis as a case study to show how we can utilize the knowledge about gene-drug interaction to help us understand how a drug can cause retroanalysis. Retroanalysis could be life-threatening. It's due to destructions of skeletal muscle caused by diverse mechanisms, including drugs and toxins. Now, what happened is that we understand there's a dramatic increase in bioplasmic ATP that is parallel with sustained elevation in cytosolic calcium concentration that lead to retroanalysis. This is a common signature of retroanalysis. The symptoms could range from myalgia to muscle weakness precede retroanalysis. So what we did was we mined the label from drug labeling, and we also mined daily mate, and we curate them. After the text mining, we curate them and identify the list of drugs. Roughly, we have 200 drugs, and then we get the gene expression data from Connectivity Map. Connectivity Map is a database sponsored by NIH and is produced by Broad Institute. Broad Institute is under MIT and Harvard. Broad Institute not only have gene expression database, but also have proteomics data. So what we did was based on that and to understand the gene expression profile, we did the workflow here is to to show you on the left-hand side to show you how the workflow of this research project. So we identify carnitine palmitoyl transferase 2 as a gene of interest. We find out this gene is the top 1% of genes that would differentially perturb by 75 drugs that cause rhabdomyolysis. And among them, 9 drugs downregulated this enzyme, with 4 of them being antipsychotic or antidepressant. Now, if we look into the biological pathway, we will be able to understand the mechanism. So we understand CPT2, this enzyme, is present in the inner mitochondrial membrane. Fatty acids have to be oxidized, utilized for ATP production through mitochondria. One of the pathways that I would like to focus on is CPT2. CPT2 genetic mutation have been can cause fatal outcome, and several states in the United States actually require a newborn to be tested for CPT2 mutation. And that is a way to help the newborn to be able to survive. So CPT2 is involved in long-chain fatty acid, medium-chain fatty acid oxidation. Through this, the enzyme CPT1 and CPT2, eventually the long-chain fatty acid and the medium-chain fatty acid become acetyl-CoA. Now let's focus on this pathway. So there's a substantial amount, there's a fixed amount of energy, ATP, come from this pathway. So let's assume that there's 1 minus X coming this pathway. Let's focus on this linear pathway. Now, eventually, acetyl-CoA is metabolized through this 3-hydroxy-3-methyl butynyl-CoA, and then hydrolyzed metabolized by HMG-CoA reductase, and to form the velolinate. This then continues to be oxidized to produce ATP. Now, the statin, we know statin can cause myalgia, muscle weaknesses, and even rhabdomyolysis. And what happened is that statin, this class of drugs, actually inhibit HMG-CoA reductase, and therefore reduce and prevent HMG-CoA to be continued to metabolize to form ATP, and that is ATP is important for muscle action. So then this is the mechanism that we can explain in terms of the effect of statin on causing rhabdomyolysis. I'm sorry, my screen just freeze. Okay, now, so then if we look further into literatures, can we find out what is there any gene drug interaction? And indeed, we find out that statin, when the patient carry genetic mutation in CPT2, such as at different amino acid location, they cause clinical phenotype that is related to rhabdomyolysis. And so one of the patients, 77 female, have myoglobinuria. And then, so those cases, actually, they gone through the so-called take the drug away and then put a drug back to give the drug patient again. So and to see, that is not purposely, okay, but the doctor actually, that's how they find out, and then they proceed to do the mutation, the genome, the PCR, and find out the CPT2 in those patients actually have mutation, and they have much lower activity of CPT2 compared to normal subjects. So those patients have the CPT2 activity ranging from 40 to 38 to 13. This is a valproic acid in these patients 47 male, 7 years old male that developed rhabdomyolysis after taking valproic acid. Valproic acid actually is metabolized and it formed valproic carnitine, and it took advantage of this pathway coming through this acetyl-CoA. What happened is that it took away the carnitine that is before the acetyl-CoA got converted and transported into the mitochondria. There's the step that I did not show here is carnitine is involved. And so valproic acid actually, when a patient take valproic acid, the outcome is depleted carnitine, and then the fatty acid will not have, the patient will not, in the mitochondria, there will not be enough carnitine available for fatty acid, long-chain fatty acid to be oxidized through the transport by CPT2. That's why we have this gene-drug interaction. Now, I would like to come back here to say that we can utilize the information based on the case that I presented earlier, rhabdomyolysis. We can even down to the gene transcriptomic data and find out what the enzyme is or the gene or the protein got perturbed. And based on that, we were able to find out that in the literature, patient had mutation, actually have CPT2 mutation, actually had experienced rhabdomyolysis when they took statin or valproic acid. So that support our finding, and then also help us to understand the gene-drug interaction. So summarize that system pharmacology approaches are useful to integrate data across systems for understanding drug safety due to gene-drug interaction. Now, the next case I would like to illustrate with proton pump inhibitors induced hypomagnesemia. So in 2011, the Food and Drug Administration informed the public that prescription proton pump inhibitors can cause can reduce serum magnesium level, cause hypomagnesemia if taken for prolonged periods of time. In most cases, patients have been taking PPI for more than a year, and PPI is a class of drugs for treating GERD, that is acid reflux. PPI or prodrug, they can be activated and then inhibit gastric acid secretion. So in approximately 1/4 of cases reviewed by FDA, magnesium supplementation alone did not improve low serum magnesium levels, and the PPI had to be discontinued. So those are very severe cases. However, there are cases where magnesium supplementation actually help. And I bring up this case study again because 2017, FDA updated information and continuing to state that PPI, prolonged use of proton pump inhibitor, can cause low serum magnesium. But then they also note that over-the-counter PPI tend to not to cause the such a serious adverse reaction. So the action is that proton pump inhibitor reduce the amount of acid in the stomach and are used to treat conditions such as GERD, stomach and small intestine ulcer, and inflammations of esophagus. So in 2009, approximately 21 million patients filled PPI prescriptions at outpatient retail pharmacies in the United States. So this is a public health issue. And so the patient who took prescription PPI tend to stay on therapy for an average of about 6 months. And that's why it's important that we find out why PPI causes hypomagnesemia. And that's what I did for my research. But I first would like to describe in terms of the sheer volume of prescription, because the class of drug we have so many prescription drugs in this class, such as esomeprazole, magnesium. So there's a magnesium in there. That's good. There's lansoprazole, omeprazole. Omeprazole were approved much earlier, and then they have a combination of omeprazole and sodium bicarbonate, lansoprazole, pantoprazole, robeprazole sodiums. There are Vibonvolt is a prescription combination product that has esomeprazole, magnesium, and naproxen in there. Then over the counters, we have all over the counter. The patient could just go into the pharmacy and pick up an OTC product. So this is a very important public health issue. We want to understand how we can mitigate this risk. Is there a way to use modeling and simulation to help answer the questions? And so at that time, because it's a public health issue, that the clinician were involved, and they first actually did the literature review. They identified 22 out of 23 literature report had adequate clinical information to assess the report. And among 17 literature cases shows that the challenge means that if you take the PPI, the drug, away from the patient and then recharge them, means that give the patient PPI again, let them to take PPI again. So 3 out of which shows recharge that can lead to that actually show hypomagnesemia. So the drug taken away, hypomagnesemia disappear, but then got recharged, then hypomagnesemia occur again. And they did not observe any obvious demographic trends, age, genders, or countries, or national diet trend. And genetic analysis for magnesium transport channel abnormality in affected patient were not performed. So that was the conclusion from the clinicians. And the signs and symptoms reported in those patients were consistent with hypomagnesemia. And also in AIRS, FDA have AIRS and they have AIR cases. In some of the cases, resolution of hypomagnesemia was seen with PPI discharges. So in other words, that is the PPI was, the patient stopped taking PPI, and then return of hypomagnesemia was seen with PPI recharging. And those patients experienced severe adverse reactions, signs and symptoms. Those are consistent with hypomagnesemia. So this is the information reviewed by the clinicians, Dr. Ethan Hausman, and we collaborate with him. So now let's look at what will happen, what is in the literature in terms of the effect of PPI. We find out that proton pump inhibitor actually can reduce distal intestinal pH. We understand stomach is acidic, in duodenum pH is around 5, and when jejunum and ileum, the pH start to increase, reach around 7 or 7.5, depends on location. Majority are around the 7 and 7.5. Now this shows, so the black one, this black navy blue represent with when the patient is on PPI, and the pink one represent the patient without PPI. So they did the pH a study in people and then divide them into groups and then check their pH after they take PPI or no PPI. So this shows that actually PPI can reduce distal intestinal pH by almost 0.5 unit. And so what is the outcome? What would this affect? How would this affect serum magnesium level? So from the literature, we find out that actually PPI can affect proton pumps that are in other tissue. And so in the pancreatic duct, that there are actually proton pump there. So the proton pump not only present in the stomach, but also is present in the pancreatic duct. And then that actually regulate the pH of the pancreatic juice into the small intestine. So the proton pump is facing this side is lumen, this side is the interstitium. And so when the patient is taking the proton pump inhibitors, they actually reduce, they actually activate the activate the pump, and then that allowed reduce the amount of bicarbonate secretion into the lumen, but then in such a way that actually increase and then decrease luminal pH. So to summarize, PPI approach drugs, the activation in acidic environment in the parietal cells in stomach, they inhibit proton pumps and then inhibit acid secretion, thereby increase pH. But they also have off-target effects. That is, activation of proton pump occur at higher pH as well, and even though at a slower rate. And that is, it was found that pantoprazole, lansoprazole, omeprazole, they have an activation half-life of around 5 hours or 1.4 hours, 1.5 hours, 1.5 hours, respectively at pH 5.1. So omeprazole and lansoprazole have a shorter activation half-life at pH 5.1. Pantoprazole have a longer activation half-life. So in the pancreatic duct, even though the pH is a lot higher, but still proton pump can be activated. And so the outcome is that they was able to see in 20 healthy subjects, 1 week of omeprazole, 40 milligram twice daily. They did not have effect on intestinal motility, but reduced the pH by 0.5 units throughout the mid to distal intestine, which is equivalent to an increase of proton concentration by 3.2-fold. So this information was used in our modeling simulation. I will show you. And so omeprazole was studied and inhibit gastric proton pumps in the plasma membrane of pancreatic duct and reduce proton in secretion into the interstitium and secretion of bicarbonate into bile duct. And that's why the pH was reduced by 0.5 unit. Here I want to mention that in the intestine, actually absorption of the magnesium took advantage of 2 routes. One is pericellular route. The other one is transcellular route. Pericellular route means that through between the cells. Transcellular route means through the membrane, and that actually can help us understand for the transcellular route is saturable. As the magnesium intake increase, it reached the constant, becomes 0 order. Now in the lumen, in the intestinal lumen, magnesium is absorbed through a TRPM6, is a transporter. In the kidney, magnesium is also reabsorbed through this transporter. So we can understand that if this transporter is affected in terms of its absorption, transport efficiency, it will not only reduce the absorption of magnesium that is present in the intestinal lumen through dietary intake, but also can reduce reabsorption of magnesium from the kidney cell in the lumen into the blood. So there's a two effects going on at the same time when a patient taking PPI. However, due to the amount of information we have, we only focus on intestine absorptions, and that's where I'm going to show you the outcome of our research. So I would like to focus on modeling intestinal absorptions of magnesium and the effect of PPI. So first, let's look at this absorption. So this is just to show you the transporters, the magnesium. There's a channel for magnesium to be absorbed. And we understand protein, the magnesium had to bind to the protein, and the protein had amino acid have side chain, the carboxylate, such as aspartic acid, glutamic acid, those are the side chain will be ionized at higher pH and bind magnesium, and then facilitate magnesium to be absorbed through this channel. And this channel can be saturable. That's why earlier we mentioned there's the absorption can be saturable. When the magnesium intake is high enough, then absorption through this route is a zero order process. So this again just kind of reiterate that when a patient takes PPI, the intestinal pH is reduced. The transporter will not have the side chain, that is an ionized side chain, to be able to bind magnesium to help magnesium to go through the magnesium channel and then reduce magnesium absorption. Now, in the kidney side, even though I do not have it in my model, but you can imagine that actually the magnesium reabsorption into the blood was also decreased. So that's recapped. Magnesium homeostasis is a balance of input absorption through oral and output is secretion through the kidney. But then the kidney will have also a mechanism to help reabsorb some of the magnesium back to the blood. So literature support the notion that PPI-induced hypomagnesemia is extrarenal. That is, we understand that the kidney part, that the majority of the evidence shows that PPI-induced hypomagnesemia is extrarenal, and so that also help us to when we did a research to set the kidney aside. But we still don't know if a patient who have the transporter mutation, what would happen. That is, the patient is taking PPI, reducing magnesium, and the original, let's say, the existing genetic mutation of in a patient that would have lower reabsorption. And we did not investigate that part. But anyway, at that time, we decided to just focus on testing. And our hypothesis was that PPI indirectly reduce intestinal absorption of magnesium and cause hypomagnesemia following a prolonged use for more than a year. And so we had to, based on this hypothesis, and conduct modeling and simulation. And the reason is indirect is PPI did not deplete magnesium, but then is affect the pH in the intestine and affect the transporter in the intestinal epithelium, and then reduce magnesium absorption. Now, a multi-year clinical trial is unethical, not possible. So the best way is to use modeling and simulation. So we published this paper focusing on simulating the effect of proton pump inhibitors on magnesium homeostasis, especially focusing on oral absorption of magnesium. So in our physiological based pharmacokinetic model, that is so-called PBPK. PBPK is an approach that is widely used in the industry, especially in the early stage drug development, when they develop formulations using the data from in vitro animal data, try to predict first dose for human to be studied in humans. And so we utilize that concept. Now that actually PBPK, this type of model is widely used in FDA to facilitate evaluation of drug-drug interaction. How would drug-drug interaction affect a drug candidate in terms of its pharmacokinetics exposure, because we understand exposure is linked to its response. And you have through a drug-drug interaction, a drug could, because a lot of patients are taking multiple drugs, especially for the elderly, when they have many diseases, then the possibility of drug-drug interaction is really high. So we are using this type of modeling approach to help us assess the risk of drug-drug interaction. Now, for this case, we use modeling, PBPK modeling to help us understand and to simulate the effect of proton pump inhibitor on magnesium absorption. So we utilize intravenous and oral magnesium pharmacokinetic data that was published in 1998. And then we use a 3-component model in terms of the systemic system. And then for the intestine, we have an absorption model, and that we include a transporter. So we have a exposure PK model. Then we also have a local, have a GI model. So here I would like to show you the GI model. So here we have the stomach, and then we have 4 steps. The drug, omeprazole, they got taken right and is not released. So this time the formulation is intact, then not dissolve, and then start to dissolve, and then continue to get into the smaller intestine. So this graph to show you from stomach to duodenum and travel this formulation simultaneously. This formulation could be disintegrate, and the drug could be dissolved and then start to be absorbed. And then in the earlier I'll show you, we have 3-compartment model for the PK. So here is a central compartment that is a circulation, and there's a peripheral compartment and 3rd compartment. Those are hypothetical compartment. It essentially to fit the plasma concentration profile. And so we have 2 compartments in addition to the circulation. And so one is, I would call it peripheral. The other one actually is deeper compartment. A lot of time, based on the drug, depends on the drug could be trapped somewhere and then redistribute and into the peripheral compartment and then get back to the central compartment. So for our case, the drug got absorbed into liver and then going to those PK compartment, 3-compartment distribution. But this is show in terms of the transit, because we want to input the transporter in the intestine, and that will help us to model and simulate the effect of PPI. So again, here, this is a diagram in terms of showing our model in terms of at the cell level, that the magnesium is absorbed through the transporter, TRPM6 and TRPM7. Earlier, I showed you TRPM6, but actually they are together, and oftentimes they are involved as in magnesium absorption, but TRPM6 is most studied in terms of its characteristic, and also is play a major role in magnesium absorption. So now this is a simulation. So we have those PK 3-compartment in the small intestine. We have so many segments from the stomach to the distal intestine. This shows you in the subject plasma concentrations of the magnesium. So this is the magnesium, the baseline, the circuit. This is the, in terms of on the x-axis is the day starting from 0 to 60 days and beyond. So this is the baseline. And so if this to test the model. So if we deplete the magnesium, so magnesium, say, the amount is decreased to 100 milligram per day. After a steady state, you reach the magnesium concentration will be lower by from 0.83 to 0.8. But if you replenish, give the model more the daily intake of magnesium to be 300 milligram per day, you can see that the magnesium level was able to go back after a while, then go back to the baseline. So this is to test the model capacity and see whether we can, without the drugs, we can reflect in terms of taking the magnesium away and then put it back, how would that affect plasma concentration of magnesium? So next, I would like to show you our simulation. The break curve represent no proton pump inhibitors. So the y-axis is the magnesium concentration in the plasma, and the x-axis is the time. And I show you the time from day 89 to 63. You want to show you the steady state after the patient is on the PPI for more than almost close to 2 months, and start to show you the steady state. With proton pump inhibitor, you can see the magnesium level decrease from around 0.85 to 0.79. Now, in terms of, in this simulation, our oral magnesium is assumed, the daily intake is assumed to be around 237 milligram per day. This is based on USDA information. We collaborate with USDA and utilize their information to understand daily intake in terms of statistics from different type of foods and throughout the United States. This was averaged to be 237 milligram per day. And assuming we understand earlier that the proton pump inhibitor can reduce intestinal pH by 0.5 unit, and that 0.5 unit is equivalent to decrease decrease the proton concentration and then reduce the transporter activity by around 3.54. And that's our assumption. We cannot detect and check on the transporter activity, but based on the reduction in the pH in the small intestine and based on the transporter in terms of their side chain, their pKa, we can estimate the availability of the side chain for transporter to bind magnesium and to be able to transport magnesium through the channel. And based on that, we deduce that the transporter activity is assumed to be reduced by 3.54. And so that was used for our simulation. Now this simulation show you the control, that is with the magnesium is with 66.3 milligram TID. That is 3 times a day. Now, this is the 6 points. This is a way of interesting thing is to show you. Okay, so the red line show you the frequency, the more frequent intake, even though the smaller doses of magnesium, but you can still maintain high magnesium, right? But then when you reduce the frequency, but they increase the dose 3 times higher in the control, the break one, you still can maintain similar magnesium profile, even though you have higher fluctuation. And this is a model, a simulating result. But if we put this subject on PPI, and we give them still maintain 100 milligrams of the magnesium, you can see that the magnesium plasma concentration is reduced. So this is a way to show you that the magnesium PPI can have effect on magnesium absorption. And then if we supplement the PPI, supplement the patient with magnesium, then we could increase the magnesium concentration. To summarize, the quantitative system pharmacology enables simulation or a strategic mitigation of PPI-induced hypomagnesemia. And so, in other words, majority of the cases, we can see that daily magnesium supplementation actually can help increase the plasma concentration magnesium to and put it and elevate the. When I say elevate, means that actually make it into normalize that. Even though if PPI could reduce magnesium serum concentration, but PPI magnesium supplementation could help magnesium to go back to the normal level. Now, I want to show you the importance of QSP. And I know this audience, we have students here, we have faculty here, and I certainly will hope that we will have an opportunity to collaborate. I want to show you in terms of use of quantitative system pharmacology in the industry and in regulatory submission. Starting in 2013, we have 1 or 2 submissions. By the time we have the workshop with ASCPT, NIH, and ISOP, that was early on in 2019, March 2019. But at the end of March, end of 2019, we have absorption. We have a submission up to close to 37. By the end of 2020, that year alone, we have 57 submission. So total, we have around 160 submission. Now, 2021, I didn't show, but 2021, we have roughly 63 submission. So it reached a plateau, but you can see that QSP is being used to facilitate drug development. In the industry, QSP is being used for drug discovery, identify drug target in terms of target engagement, and to predict effect, and to design the first dose for human, but then also used for phase 2 study design, phase 3, even post-marketing. And I want to show you application of QSP across different diseases area that we have majority are in the cancer area. So including the solid tumors, oncologic diseases, and hematologic malignancy. But we also have QSP application in neurology, Alzheimer's diseases, and other diseases. And also another big class is cardiology and nephrology. Now, in terms of application, you can see amount up to 2020, we only have one case that one submission applied to design dose for monkey study. The rest to design study in humans and distribute from phase 1, phase 2, phase 3, and post-marketing. And then I had to apologize. In terms of distribution between NDA, BLA submission, and PIND or IND, majority are in the drug development phases, that is PIND, IND stages. And thank you. And I hope that I can help answer the question. I want to emphasize is that industry need scientists, young scientists, and especially engineers such as PAASE, such an excellent institute. And I'm sure there are a lot of very talented young scientists out there. And I hope you can join QSP community and help to grow QSP and use that for drug discovery and drug development, especially in drug development. And I understand in Asia, especially drug combination, utilize the drug that have been approved before, but put them together, we can design different products and to help treating diseases. And thank you so much.
Thank you so much, Dr. Bai.
Thank you.
So for the Q&A portion, I will give the floor to Dr. Stephanie Macalino.
Thank you very much, Dr. Bai, for that, for the encouraging words to the Filipino scientists and the young scientists that are currently in the room with us. And thank you very much for that fascinating talk. Okay, so now the floor is open for questions from the audience.
Dr. Bai, I do have a question.
Mhm.
Can you give us details of the simulated experiment, the ones that with the graphs that you showed us? Is this computer-based that you put in the assumptions?
So yeah, so based on the assumption we designed, certainly we use the software that is available. We use GastroPlus, and that's available to us. But we based on the information we gather from literatures, we manipulate the model and to generate the graph. Currently, and I would like to mention this because there are so many young scientists, talented scientists here. Currently, there's a need to simulate variability. What I have is an average subject. But if you can simulate variability and predict variability and separate and be able to identify responder from differentiate responder to a drug treatment from non-responder, and to be able to have precision, so individualize, identify patient that could respond to a drug, that is very needed now. That is we need young scientists to put in effort into develop methods, standardized methods that is credible, that we can utilize to help in terms of designing clinical trial, to help differentiate responder from non-responders, and also to help simulate and predict variability that can actually help in terms of study design, in terms of statistical power needed, the number of subjects needed during the clinical trial. But not only that, okay, I forgot to mention, I didn't mention, but in my published landscape analysis, actually sponsor industry is proposing using QSP to conduct virtual trial to ask us to waive the trial needed. There's a need for helping finding cure to treat rare diseases. So imagine that we only have 10 patients worldwide. If we can create digital patient, digital twins, okay, let's have imagination. If we can create digital twin for rare diseases, and then use them to do clinical trial, that will really help a lot, because a lot of time the drug developer will have to conduct additional trial after initial approval, but then there are requirements for post-approval, PMR, PMC, they need to conduct study, they need to recruit more patients. And so this is where engineering can come in to help, to create digital patient to help conduct clinical trial. And I hope, I'm sorry, I just want to make sure that you heard the message that join the QSP community. And I know the Zoom is so easy, right? We can do research together without, even though distance is so far away. There's ISOP has a QSP community that in the past 2 years, they have organized QSP symposium for students. And I will be very happy to help to make connection if next year, they usually around May, if they have the symposium for 2023, I will encourage you to submit your poster and to meet with the panelists and to ask questions about the opportunity, about the needs, And then share your research ideas through the poster presentation to let people see what you are doing.
Okay, all right. So any other questions from the audience? So as mentioned, in the chat box, so you may place your questions there and I will be able to read that out to ask our speaker. All right, so we already have a question here. Is it recommended to remove over-the-counter PPIs to avoid hypomagnesia?
No, no, I mean, PPI over the counter is so helpful for millions of people. Then the thing is that they actually have most recently on FDA website 2017, they have a statement that over-the-counter PPI have lower strength, and so they did not see the safety concern as compared to prescription PPI. But certainly, it's very important to be aware that when you are taking OTC, if you are more sensitive, we don't know individual patient, they may have the transporter mutation, right? So just be aware of the issue, and you want to increase intake of magnesium up to make sure that you have that. And if you experience any side effect, then you want to check it out, whether it's because of hypomagnesemia, and then you want to have a supplementation. And I actually talked to people in Japan. They actually, in their tap water, somehow they put in more magnesium, magnesium in there. And they said that don't worry. In Japan, we have that. We have more magnesium in the water. But anyway, so that I would like to share that with you.
Thank you for that. Next question. How has QSP impacted drug development, especially FDA requirements to be able to predict potential serious side effects in new me-too drugs that are widely prescribed?
Okay, so there are QSP has been used to predict liver toxicity. That is one application. The other one is there are drugs that could deplete iron. So they're having QSP used to predict iron concentration for drugs. And when you say me too, can you rephrase that? Are you talking about generic drugs?
Yes. So it's already been released to in the market, but then other pharmaceutical companies create the same drug, maybe different formulation.
Right, you can use a PPI if there is a safety concern that you want to find out how you can mitigate using model and QSP to help address that questions. But in terms of in drug development, from the new drug side that I have seen is that QSP has been used to design from IV to sub-Q, from a single dose to step-up dosing. You understand step-up dosing, so you say original is 50 milligram sub-Q, but then you change it to 30 plus 30. And then you want to reduce release of cytokine, because a lot of immune checkpoint inhibitor or monoclonal antibody for cancer, they can induce cytokine release. So in drug development stages, they can be used to help design dosing regimen. Currently, there's a goal in terms of help identify responder and identify non-responder to help clinical trial design. And that is still a work in progress. I have not seen much success because I'm the lead on the QSP, so I'm very aware of the application. And I hope I answered your question.
Okay, thank you very much for that. I think we just have time for one last question here. Question from Miss Cecilia Jimeno. Just curious where most of the activity in research on QSP is happening in the US. Is it mostly in the industry, which are developing drugs, or in independent academic laboratories where scientists are working with regulatory agencies or pharmaceutical companies. So this is quite novel. That's why they are trying to visualize where the activity is coming from.
I'm sorry, I didn't. Is that a question for me?
Yes, yes. So where for QSP, which one? So rephrasing this question, which one has the most activity when dealing with QSP? Is it the industry or is it the academic laboratories?
Actually, I would say it started from industry. Long ago, Antero studied the notion of creating a platform model for a disease. And the company didn't survive, but all the scientists in the company just went for a job in the industry and create cluster. So Pfizer, Genentech, Bristol Myers Squibb, Roche, those are big companies that need QSP scientists. They have a huge group. Not only that, I'm sorry, Merck maybe not as active, but smaller company continue become aware of the utility, and so they actually hire scientists or they hire CRO. So there are CRO in India, in Russia, who specialize in helping people creating QSP model and then do simulation for industry. So where the jobs are, I would say a majority are in industry, but academia, they also need QSP modeling. NIH does support multiscale modeling. NCI is very interested in multiscale modeling. DARPA, Defense, and also Department of Energy, they also need QSP modeling. And they use a term called multiscale modeling, but also there's a tremendous amount of interest in terms of digital twin. Now in regulatory environment, we are catching up, I should say. But in CDRH, they do have a huge group of modelers. But if you're interested in integrating pharmacology, because early on I learned from the video that actually at PASSE, you also have laboratory pharmacology. So integrate, actually at this institute, you have all the component available for conducting QSP modeling. In terms of more focusing on drug development, I understand there are outstanding scientific accomplishment at PASSE, but what I'm alluding to was collaboration in terms of promoting drug development and development of combination product. That's what I'm hoping to promote, especially for scientists in Asia, to utilize your talent and to produce product for the public. If we cannot do drug discovery first, but drug combination, using me-too product and combine them, that could help the public. Absolutely.
Okay, thank you very much for that. I think that's all the time we have for question and answer. If you do have any more questions, I'm sure that Dr. Bai will be open to any communications from any of us. Right, so let's take the time to have a Zoom feed group picture here in Zoom.
Thank you so much. By the way, if you have questions, if you want to make connection with ISAP, the QSP Student Symposium, just send me an email. Eileen has my email, right? Just don't hesitate. Angie also has my email address. Just contact me and I will be very happy to facilitate collaborations and help grow, help the young scientists to interact with the scientists here. And if there's an opportunity in terms of for you guys to visit FDA, to come as a fellow, to join us, I will be very happy to facilitate.
All right. Dr.
Once
Lau?
again, thank you for joining us, and that ends our webinar for today. Thank you again, Dr. Bai.
Thank you very much, Dr. Bai.
Well, thank you all, and thank you so much for this opportunity. And I hope we can grow QSP for drug development together. And thank you. Take care. Okay.
I'll end the meeting in a count of 3, 2, 1. Bye.